Good morning, dear students. Today we are going to start our section with the heading Dorsal Ventral Patterning in the oocyte. In the last video, we have just learned about the development of anterior-posterior polarization in Drosophila. And those who didn't watch that video, please go and watch that video. Only then you can understand as you watch this section. So let's start. Here I just drew the picture of Drosophila egg. Here we can see the follicle cells, the oocyte, then all these nerve cells. And in the former section, we have just seen that during the time of development of anterior posterior axis, the nucleus has moved to the more near to the terminal follicle cells, somewhat here, terminal follicle cells, and results in the posteriorization of these follicle cells. Okay, and also we have learned that as the cytoplasmic components of the nerve cells gradually move towards the oocyte, the volume of the oocyte will increase, or it keep on increasing. And among that cytoplasmic contents, it includes so many mRNAs that is very significant in the further development process of Drosophila. All those things we have seen in the last video. And during the dorsal ventral patterning, the first thing that is happening is that the nucleus, it moves towards the dorsal side of the egg. Okay. And because of that movement, it now closer to the dorsal follicle cells. Okay. And for explaining everything so clearly, I am going to redraw only this portion in the next slide, and I will continually explain. So that portion I redrew here. Here we can see this much area. This is the oocyte, and this space is the pre-vitelline space, and this membrane is the vitelline membrane. These are the follicle cell, and all these portions are part of dorsal side of that egg. Okay, clear? I think you are familiar. You understand about which part I am going to explain. So we have seen that the nucleus move closer to the dorsal side. And in the last video, we have seen that an important mRNA, that is Gurkhan mRNA, it is translocated to the oocyte from the nerve cell. And here also we can see the Gurkhan mRNA, and it locates between a position between the nuclear membrane and that oocyte membrane. And then this Gurkhan mRNA undergoes translation and produce the Gurkhan protein. What is forming? Gurkhan protein. And this Gurkhan protein, what its next fate in the follicle cell, like in the case of that posterior follicle cell, here also present torpedo receptors, a product of torpedo gene. And this Gurkhan protein, I'm just explaining the balance portion, taking a single Gurkhan protein. But you have to understand that this mRNA undergoes repeated round of translation and producing enough number of Gurkhan protein. Okay. So anyway, Gurkhan protein go and bind with the torpedo receptors. Okay. And this binding, it activates, it initiates, or it prompts this dorsal follicle cells to undergo dorsalization. This binding of Gurkhan protein with the torpedo receptor that present in the dorsal follicle cell, because it's a dorsalization. So that is the process that lead to the dorsalization. Okay. Along with that result, along with that, one more change is happening. This activated torpedo receptor, it can inhibit what? Inhibit the transcription of pipe gene. pipe gene is present in the follicle cell nucleus, and as because of the lack of enough space, that's why I draw it outside. And when you understand that, this activated torpedo receptor inhibits the pipe gene. So, because of that inhibition, ventralization is not possible to happen for these follicle cells. From this, you understand that this pipe gene is very important, crucial in the ventralization of the follicle cell. So let's see how it is happening. In the next slide, I have just drawn only that ventral portion of the egg. Here also we can see the ventral follicle cell, and this is the pre-vitelline space. This is the pre-vitelline membrane. Okay, and here, as there is no Gurkhan protein or torpedo receptor, here that 5 genes transcription happens, and a pipe protein is forming here. And in this vitelline membrane presents some other protein. What is its name? Noodle protein. What is the name of that protein? Noodle protein. And this pipe protein, it goes and bind with the noodle protein and activate this one. Okay. And a few hours later, after the development, that embryo or that oocyte secretes some serine proteases, mainly 3 type of serine proteases. We'll see the case of each one separately. First one is a gastrulation defective protein. And this protein is secreting to the pre-vitelline space, and it comes and binds with the activated noodle protein. And how this serine protease get activated? It get activated because of a peptide cleavage. And after activation, then we need to mention about the second type of serine protease. What's its name? Snake. And this also secreted by the embryo to the pre-vitelline space, and it comes and bind with the activated gastrulation defective gene, and it also get activated through a peptide cleavage. And then came the third serine proteases, that is ester. This ester protease is also secreted by the embryo to the pre-vitelline space, and that also in the same way come and bind with the activated snake protein, and it also undergoes a peptide cleavage and become active. So how many serine proteases we have seen? Gastrulation, defective gene, this one, snake, sorry, not gene, protein, snake protein and ester protein. Okay. Then finally, there is one more, spacil protein. This activated ester protein, to this ester protein, this spacil protein come and bind, and this also undergoes cleavage and got activated. Okay. Then in the oocyte membrane there present a toll protein, and this activated spacil protein, it goes and bind the toll protein and activate this one. Okay, and as this toll receptor got activated, then to there came to another protein, 2 protein and pelle protein. Pelle protein, this one. Actually, this 2 protein is a carrier to fetch this pelle protein to here. And this pelle protein is a protein kinase. Okay. And as the after 19 minutes of fertilization, the oocyte will be in syncytial condition. Syncytial condition, we know that there will be so many nucleus without individual cell membrane. Okay, so syncytial condition. So, in this syncytial condition, a dorsal, a dorsal protein is forming here. When it is forming? After 19 minutes of fertilization, this dorsal protein is forming here. And this dorsal protein is not found single. It always seen associated with the cactus, cactus protein. So, this cactus is what it is doing? It is suppressing. It is controlling this dorsal protein. And as this Pele protein become activated, it adds or it phosphatizes the cactus, and because of that phosphatization, the cactus is separated from dorsal, and the dorsal then can enter into the nucleus that present in the ventralmost region of that embryo. Okay, next we are going to see what is happening inside the nucleus as the dorsal enters inside that nucleus. So here we can see the dorsal that is entered inside the ventral nucleus, and it acts as a transcription factor and results in the transcription of certain genes. At the same time, it inhibits the transcription of some other genes. Which type of gene get transcribed with the help of dorsal? Those gene that is responsible for the ventralization of that follicle cells, that only get transcribed, and those gene that is responsible for the dorsalization will not transcribe. It is suppressed by this dorsal protein. So in this way, the dorsal ventral polarization is happening in the Drosophila. Okay, I hope it's very clear to you. Thank you.
I'm an icon, man. Ain't my girl about a 10. I should fuck her in a Benz.